We continue the series on DNA replication, and this is a continuation on the prokaryotic elongation video. And in this video specifically, we will discuss the process of DNA synthesis by understanding its mechanism. This will apply to both prokaryotes and eukaryotes. We will discuss some very specific questions, and these are pretty straightforward questions. We will start by understanding how DNA polymerase differentiates between dNTPs and how dNTPs are further differentiated from rNTPs. We will also try to understand how base pairing of the incoming dNTP is established with the template. And when the base pairing is established, we will dive into the process of phosphodiester bond formation, which, as you may know, is essential for the backbone of the DNA. And finally, we will understand how DNA polymerase eventually moves or translocates after adding the dNTP. All of this will help us understand how DNA synthesis actually works. To get a structural functional relationship understanding of the mechanism, I will take the example of the prokaryotic DNA polymerase, more specifically DNA polymerase I. We talked about the DNA polymerase I in the elongation video, where we discussed its importance in the context of the Klenow fragment, which has the 5' to 3' activity that makes the DNA. The other activity of this enzyme is the exonuclease activity. These two make up the C-terminal domain of the DNA polymerase I. More specifically, we will focus on the Klenow fragment part of the C-terminal domain, because that is the important part involved in the DNA synthesis. And along with Taq polymerase, it happens to be the most widely studied polymerase and has contributed immensely to our understanding of DNA synthesis. Before we jump into the details, I want to simplify the entire concept. First thing to note is that the polymerization occurs towards the 3' end direction of the growing strand. Let's get an intuition of the process by sketching out a template DNA strand, which has phosphates in its backbone. As we noted in the elongation video, the process of DNA synthesis starts when the RNA primer is laid on the template strand. So here is the RNA primer. Just to be specific, the uracil exists because it is an RNA primer. And these two strands are bound by hydrogen bonds. 2 between A and T, or A and U, and 3 between G and C. The important thing to keep in mind is that the final base has a free hydroxyl group at the 3' position in its sugar. Okay, so that's the template. Now if we look at the substrate, the dNTPs, we have 4 of them, and they all have 3' hydroxyl groups and 5' triphosphates. For each reaction or synthesis step, we need one of them. So there is a 25% chance that each one is sampled for a given reaction. Given that we have a cytosine in the template strand, a guanine will be used in this particular reaction step, because it is the only dNTP that will fit the polymerase active site, and properly base pair with the cytosine on the template strand. In the process of synthesis, the DNA polymerase will try to attach the hydroxyl of the previous sugar to the phosphate of the incoming dNTP. As a result, the remaining 2 phosphates are released as byproducts. This bond that is formed between the hydroxyl and the phosphate is called the phosphodiester bond. And this again leaves the hydroxyl available for the next dNTP to be added, just as the previous hydroxyl existed. So in this next step, the cytosine will be used because there is a G in the template strand. And in this way, the DNA polymerization will continue till the end. In this simplified view of DNA polymerization, all I want you to take away is that the direction of 5' to 3' comes because the hydroxyl exists at the 3' end, and that is the only place to add a new dNTP. Now we're ready to take a deeper dive into this subject, and we will understand the synthesis by looking at the enzyme which does this function, and a specific example will be the C-terminal domain of DNA polymerase I, which, as we have said, has the Klenow fragment, and this exodomain. The polymerase function is well conserved into the eukaryotes. Therefore, you can also apply this same analogy to the eukaryotic DNA polymerase. Let me quickly sketch out the rough outline of the C-terminal domain of DNA polymerase I. The top left of the structure has multiple alpha helices in its region, and these are often referred to as the fingers of the polymerase. And this top right side is called the thumb domain, and this middle portion is called the palm of the DNA polymerase. The bottom side of the structure, the wrist portion, if you may, is the exonuclease domain of the C-terminal domain. So this entire thing is like a hand or a baseball glove. We have seen this same structure when we talked about the RNA polymerase when we discussed transcription. Our main focus will be on the top bigger portion, or the Klenow part specifically, and it will not focus on the exonuclease domain. One contrast to note is that the palm domain is largely composed of beta sheets, which help accommodate the DNA. The important thing to note is that the palm domain contains active site of the DNA polymerase, and this active site contains two divalent ions, which are usually magnesium or zinc. So when a single-stranded template comes at the palm domain, it is bent at the palm domain. And the organization of the DNA is such that the DNA polymerase is recruited at the 3' end of the RNA primer. We have seen this already in the elongation video. You can also think of this RNA primer as the daughter DNA, because it is the same thing for our understanding. The bending of the DNA is important because it exposes the template for the dNTP addition. Okay, now we're ready to answer this first question. To get to it, we'll have to focus our understanding on the upper half of the Klenow, specifically on the finger domain alpha helix. In the alpha helix, there are tyrosine, lysine, and arginine amino acid that will become very important. Another thing that I want you to keep in mind is this bending of the template, which exposes the template to polymerization. Essentially, the exposed template is ready to accept a dNTP. Even more specifically, the bending causes the minor and major grooves to become accessible by the active site of the DNA polymerase. And as a result, the few bases around the free 3' hydroxyl of the primer takes on an A DNA form. This is in contrast to the B form DNA in the remainder of the double-stranded part. All of this is occurring at the active site present in the palm region. The accessibility is initiated by these 3 amino acids in the finger helix, where tyrosine plays a major role in contacting the DNA and the dNTP, and lysine and arginine mainly contact the dNTPs. This bending and accessibility makes the polymerase accept a new dNTP, which can be sampled, and if it is the correct one, the polymerase will use it. So now our focus will shift to this smaller frame, where we will zoom into the active site dynamics of the DNA polymerase. This state of the DNA polymerase, where the dNTP hasn't yet been confirmed for addition into the daughter strand, is called the open configuration of the DNA polymerase. And when the finger domain becomes accessible, what we mean to say is that it moves down towards the palm region. And this is about 40 to 46 degree movement. Some articles may say that it moves 30 or 60 degrees, but keep in mind that the reference for the movement can be different. The point being that there is a movement in the finger domain. It is not as important to know how much. The movement occurs when tyrosine responds when it comes to the contact of a dNTP. Let's understand this in context of the template DNA that is being bent at the palm region. So let's assume that the next base to be replicated is this cytosine. This means that the next dNTP to be added will be a dGTP. So this move in the finger domain will occur in response to the correct dNTP when it is being positioned against the cytosine on the template strand. So essentially, in the sampling for correct dNTP, a fit in terms of proper hydrogen bonding will cause a sustained move in the finger domain. And now the G and C can properly hydrogen bond. So to emphasize the point, there is a stacking interaction by the tyrosine of the finger domain, which pushes the dNTP against the base on the template strand to be copied. And of course, some other weaker interaction like van der Waals interactions are also at play here. So let's complete the picture of the GTP here, since it comes with a sugar and 3 phosphates. And this picture is of course not complete without the daughter strand, which has the 3' hydroxyl ready to accept the GTP. The proper positioning of the correct dNTP is also mediated by lysine and arginine amino acids on the finger domain, as they make weaker but important contacts with the beta and the gamma phosphates of the dNTP. So now we're starting to see how these three residues are essential in this process. So now the stacking interaction is stabilized with the help of hydrogen bonds between the two bases. Instead, if we had A, T, or C instead of G, the stacking will still work, but it would fail to hydrogen bond with the cytosine. And the arginine and lysine will also not interact as efficiently. And these three dNTPs will keep getting rejected. So for a cytosine, only a dGTP will work. All these stabilized interaction with the correct dNTP is what establishes the closed conformation of the DNA polymerase. Now let's see what is so special in this closed configuration. When this closed configuration is triggered, the active site is engaged properly. This has to do with the divalent ions I mentioned a while ago. So now, with the correct dNTP at the POM region, one of the divalent ions makes contact with the phosphate of the dNTP, and the other makes contact with the hydroxyl of the daughter strand, to which the new dNTP will be attached. Alright, we have answered a bit of this question about dNTP discrimination. To recap, the finger domain simply moves into the POM site, and if the dNTP is a correct fit, the polymerase goes into a forward equilibrium to a closed state. The second question on our list was regarding the base pairing of the bases. Again, the answer to this is that the finger domain, which samples the dNTP via stacking interaction and assesses if the hydrogen bonding is favorable or not. The favorable bonding is then confirmed via proper contacts of the finger domain and the divalent ions. So the simple answer to this question is the finger domain dynamics. We haven't touched on this rNTP part of the question. We'll come back to it in a short while. Now that we have a closed conformation, we're ready for the next question, which is, how is the phosphodiester bond made? Once again, our focus needs to be on the finger domain. This domain that we have been referring to again and again is called the O-helix domain. Let's just fill out the important pieces once more. To understand how the phosphodiester bond is made, we have to shift our attention onto the phosphates of the dNTP. And on the other side, the bond will be made with the growing daughter DNA strand, which has free hydroxyl group. The reaction starts with the divalent ions interacting with the dNTP and the hydroxyl on the daughter DNA strand. For your information, these ions are not floating around in the active site. They're held together via specific amino acids, like aspartic acid residues in the POM region. So what is the mechanism here? Well, the first metal ion, let's call this metal ion B, as you see here, interacts with all three phosphates of the dNTP. And this works to neutralize the negative charge of the phosphate, and it makes it inert. And these phosphates are usually named alpha, beta, and gamma. The other metal ion, let's call this metal ion A, interacts with free 3' hydroxyl group and weakens the hydrogen bonding of the OH. Essentially, it works to take away the hydrogen. As a result, the oxygen here takes on a radical form. So this oxygen is ready for a nucleophilic attack on the alpha phosphate of the dNTP. When this occurs, the beta and gamma phosphates are released as byproducts. And this bond that is formed between alpha phosphate and the oxygen radical is called the phosphodiester bond. So the process to make the phosphodiester linkage involves this beautiful metal ion catalysis reaction. And now we can go back to this question of discriminating against rNTPs. If this dNTP was instead an rNTP, it would have a 2' hydroxyl on the sugar, and that makes the sugar a bit bigger. And the cavity or the active site where this sugar docks will not be able to fit this rNTP because of its bigger size. And this will further destabilize the stacking interaction if the polymerase tries to work with it. And all these contacts with the metal ions and the lysine and the arginine will not be able to work together. This then leads to the rNTP being destabilized and kicked out of the reaction. So in contrast to dNTP discrimination via the finger domain, the rNTPs are sampled via the POM region. So now that we have added the dNTP into the growing DNA strand, now the DNA polymerase must move forward by one base to add the next dNTP. So how does that happen? Let's just quickly complete our picture of the phosphodiester linkage reaction. This alpha phosphate and the oxygen together form the important bond. After the bond is formed, the finger domain, the O helix, hinges back and takes away the phosphates. This pyrophosphate, this byproduct, is then released. Now, the finger domain looks like it is again in the open configuration. Now, one hypothesis about the energy required to move this enzyme is possibly that the pyrophosphate released, or the phosphate bond breaking, provides the energy for translocation of the enzyme. Turns out there is another prevailing hypothesis, which involves the concept of linear diffusion, which states that because of this backward movement of the finger domain, the helix, or the fingers, act like a spring. So, in sort of a bigger picture, all this means is that when the dNTP binds the finger domain closes. The configuration goes from open to close, which leads to phosphodiester bond formation if the dNTP is correct. And after the bond formation, the finger domain retracts back. So this movement or oscillation is similar to that of a spring, which forces the polymerase to move with one base copied at a time. Apart from palm and finger domain dynamics, each time the polymerase adds the dNTP into the growing daughter strand, the thumb domain repositions the template via some sort of rotational force. The thumb domain detaches for a short split moment and reattaches in a new position. The exact mechanism underlying this movement are still being discovered, but researchers suggest that it is a mechanism involving some combination of this spring-like oscillation and the pyrophosphate release that helps in the polymerase movement.